Assalamu alaikum. I am Dr. Muhammad Ali Rabbani, and today we are going to discuss the development of ear. So, by the end of this session, you should be able to describe the embryological origin of different parts of human ear, be able to explain the differentiation of otic placode into membranous labyrinth and its further parts, identify the pharyngeal derivatives contributing to the ossicles and associated muscles. And we will discuss the key stages in development of external ear and correlate this whole developmental process with common congenital anomalies. With this, let us dive into our first topic. So, before starting the lecture, I wanted to show you what are the parts of the ear and where do we want to get to by the end of this lecture. So, the ear consists of three parts. The external ear consisting of pinna and the meatus, correct sound collecting organ. The middle ear having the tympanic cavity and the ossicles. It is conducting sound from eardrum to the inner ear, and the inner ear, it's actually the sound sensing organ. Here the transduction occurs where the mechanical sound waves are converted into electrical impulses. Also, additionally, inner ear also contains the operators for equilibrium changes. So this is the external ear consisting of pinna and this tunnel auditory meatus. This is the middle ear, the cavity containing malleus, incus and stapes, and this is the petrous bone and it contains this bony labyrinth that further contains the membranous labyrinth. So membranous labyrinth consists of these parts. It consists of the sound sensing organ cochlea. Here the mechanical signals are converted to electrical signals. There is saccule, otricle and saccule for detecting static balance information and linear acceleration, and three semicircular canals roughly at right angle to one another to detect angular acceleration. Then there is this endolymphatic duct ending in endolymphatic sac. This is for the here the endolymph is absorbed here, and there is this vestibular duct or cochlear aqueduct. Cochlear aqueduct. Here this connects. This allows the perilymph to communicate with the cerebrospinal fluid. We will see how this organ of Corti is formed. This is 1 ton of cochlea shown. This is the bone in the center called modulus, and here is osseous lamina, bony part of this lamina in which there is spiral ganglion shown here. This part, this is the actual cochlear duct. This is the membranous labyrinth part of the cochlea, and it contains this organ of Corti consisting of hair cells, supporting cells and the tectorial membrane on top of them. And this is the scala vestibuli and this is the scala tympani filled with perilymph. So this is filled with endolymph. We will go into the detail further. So this is where we want to end up by the end of this lecture. And one quick nugget from the development of head and neck that we will be encountering is the pharyngeal arches. As we have studied in the lecture of development of head and neck, that in the development there are these pharyngeal arches that are developed. So there are 5 pharyngeal arches, 1, 2, 3, 4 and 6. And each pharyngeal arches, if we cut along this line and see from the front, you will see 1, 2, 3, 4 and 6 arches. These are the raised parts. Each arch will have its own artery, its own nerve and the cartilage part that will give rise to certain bones. So in the head and neck, we study what structures are formed by each of these skeletal components, not going into the detail here. On the outside, between this is one arch, this is the second arch, in between them, this is on the outside, this is pharyngeal cleft, this is another pharyngeal cleft, this is another pharyngeal cleft. On the inside, this is pharyngeal pouch, this is pharyngeal pouch. So as you can see very well, see the pharyngeal clefts are lined by the covering epidermis or epithelium. So this will be ectodermal, lined by blue here in this picture of Langman, and in the inside this is covered by endoderm of the pharynx. So whatever structure is developed from the pharyngeal cleft will be ectodermal in origin, and whatever is from the pharyngeal pouch will be endodermal in origin. So these are the pictures, not going into the detail, but these are the parts which we will be discussing, like the first cleft giving rise to the external meatus, first pouch giving rise to the auditory tube and tympanic membrane. So I wanted to just give a little heads up of where we are going to be going or where we are directed in this lecture. So let us start with the development process. This is the embryo roughly at the end of third week at the day 21-22, and as you can see in this area there is a thickening of the skin is formed that later invaginates. So this is called otic placode, the thickened part is. So let us start with the inner ear. So this thickened part is called otic placode. It's usually developed at day 22 and it is opposite to the hindbrain or rhombencephalon. Then as the time goes, this thickened part, it dips inside, it becomes otic pit. We say the otic placode invaginates, then it becomes otic pit, and as it goes in, it starts to pinch off, and gradually it's pinching off completely from the surface, so it becomes otic vesicle, also called otocyst. So this is the part which will ultimately give rise to the inner ear. It's all parts in the membranous labyrinth, and if you can appreciate here, let me zoom in, it also gives rise to the cells that are going to form the nucleus, or that are going to form the ganglion of the vestibulocochlear nerve or statoacoustic ganglion. So this otocyst, this immediately starts to differentiate, and it differentiates into a ventral circular portion and a dorsal auricular portion. This ventricular circular portion is ultimately going to form saccule and the cochlea, and this portion is ultimately going to form the auricle, semicircular canals and endolymphatic duct. So these are the structures that are going to be formed by this. So this is the ventral, this is the dorsal part, and as they develop, the connection between them constricts to become narrow, and this connection is called auriculocerebellar duct, and this duct is gives the connection of the endolymphatic duct terminating in endolymphatic sac, and that is why in some embryology books you will find that otocyst will be divided in some books into three parts. It will be divided into circular, auricular and endolymphatic parts because it does not nicely connects with, gives preference to any one of the following. It is connected in between them to the auriculocerebellar duct. So let us focus on the saccule part. Roughly beginning in the 6th week, the circular parts develops an outgrowth, outpocketing roughly in the 6th week, and that starts to grow, and that is the future cochlea, and that goes on to lengthen, and it continues to grow until it forms about 2.5 tons. Some books say 2 3/4 tons. So this is the duct that is going to form the cochlea. This is penetrating into the mesenchyme. As you see, we are fairly early in the developmental phase. We are in the 6th week. Till now, there are no bone and no even cartilaginous modulus formed for the bones of head and neck. So it is developing into the mesenchyme where future petrous part of temporal bone will be formed. Later, that will become cartilaginous. As the cochlea grows and it makes it turns, the connection between the cochlea and the saccule, it narrows, and this is called ductus deoniens. Now this is a cross section of the cochlear canal. First let us talk about the surrounding mesenchyme. Then we will see inside what is happening. So surrounding mesenchyme, it is growing inside, it gives signals, and the surrounding mesenchymes begin to develop into cartilage. It starts to differentiate into cartilage. This is called cartilaginous shell. This cartilaginous shell gradually starts vacuolization, and the gap starts to form above the cochlea and below the cochlea too. And as the more and more vacuoles are made and they unite, they form a single big cavity. The cavity that is above the cochlea is called scala vestibuli, and the cavity that is below it's called scala tympani. This cochlea The part that differentiate or the part that separates the cochlea or scala media from the scala vestibuli, this is called vestibular membrane, and the cavity of the scala tympani, it's separated from the scala media through this membrane. This is called basilar membrane, and this is the basilar membrane that gives attachment place for all these sensory hair cells and supporting cells. So this is the organ of Corti that is going to be formed, and this is supported by this osseous lamina of the modulus. Modulus was the cone-shaped bony projection here and around which the cochlear tuns were formed. On either side, the cochlear tuns were formed around it. Cochlear shape can be understood like this. It is a snail shell, just like a snail shell. It starts with a big turn from the bottom. It makes a complete turn, then makes a second complete turn, and then makes a 3/4 turn above. And each turn, in one turn, you can see there is the bone extension of the bone, that is ossicles lamina. This is the cochlea or scala media. This is the scala vestibuli. This is the scala tympani. In between them, there is this bony pillar. This is called the modulus. It can be thought of like this a Phillips screw. Like it is wider at the base, it is narrower at the top, and it has this bone, this projection, this thread going on. So this thread, like this one. This is a spiral osseous lamina in which there is this spiral ganglion is like there too. That is why it is called spiral ganglion. So this is about the cochlea. Now let us see what happens to the actual cochlear canal. Initially, this is a layer of simple epithelium, simple cuboidal in the beginning, and as it starts to grow, it becomes columnar, and then more cells are formed. The basement membrane is lying on the outside, and then the cells, as they continue to grow, the part that is lying on the bony projection, that was lying on the osseous lamina, this develops into spiral limbus. This develops into spiral limbus. It rests on the top of the bone. Here you can see this is the part of the bone that is here, like this. This is the part that will develop into the bone. As you can see in the purple, you can see this bone. So this is the bone, and this is the spiral limbus that is forming. So this is the spiral limbus that is forming, and it is lying on the top of osseous spiral lamina. It does not have any sensory function, but it has a strong footing on a bone, and it gives anchoring place for the tectorial membrane, non-cellular membrane of extracellular material that lies on the top of the rest of the cells. Then there is a gap that is called spiral sulcus, and these cells, so this was the inner ridge of cells that was giving rise to the spiral sulcus, and this is the outer ridge of cells or neuroepithelial cell that is going to give rise to the hair cells. So there are two types of cells that are developed. So there is one, this is the inner hair cell, and these are the outer hair cells. And as you can see by their name, they have hair on the top, as you can see here, that are embedded in the tectorial membrane there. So these are the hair cells, and these hair cells are supported by these supporting cells, pharyngeal cells, and there are pillar cells, and there are other cells too, but we will discuss that in histology lectures. So these are the hair cells that are formed. There is one layer of inner hair cell and the 3 or 4 layer of outer hair cells. So these are the one that are majorly sensory cells, the inner hair cells, and these are the one that technically they help to adjust the sensitivity of the inner hair cells to the sound. They help to amplify the sound. They contract and relax to pull on the tectorial membrane to help in the inner hair cells. So this is the organ of Corti, consisting of the basement membrane and the consisting of these sensory hair cells and the tectorial membrane above. And when the sound waves travel through scala vestibuli, at the point where the basilar membrane natural frequency resonates with that of the incoming sound, that part of cochlea vibrates, and when it vibrates, this part vibrates because it is resting on a basilar membrane, and this part and tectorial membrane remains firm because it is sitting on a bone. So when they occur, the hair cells they move against the tectorial membrane and they give rise to the origin of the signals. So this was about the development of cochlea and the circular portion. Now let us move on to the otrical portion. So it starts again in the 6th week, and in the 6th week the flat outpouchings roughly at 90 degree to one another starts to develop. As this outpouching grow, the part near the periphery it remains open, patent, and the part in the periphery in the center, the walls oppose one another, and that parts oppose one another, they obliterate the cavity between them and they ultimately get absorbed. And the part in the periphery, this becomes canal. So this becomes the semicircular canals. Each semicircular canal is roughly have one narrow end and one expanded end. This is called crus non-ampuller, and expanded part is called crus ampuller. And this crus ampuller is the part where the sensory cells of semicircular canal are present. And the crus non-ampuller of two superior and posterior semicircular canals, they fuse and open with a common opening into the otricle. So otricle will have 5 openings entering into it. And the sensory parts of the semicircular canals, these are in this crus ampuller, are called crista ampullaris, and the sensory component of otricle and saccule are called macula acoustic. And the further their hair cell formation, their signals and otolithic membrane, all of that will be discussed in the lecture of histology. We'll leave it at here. So these are the parts which in response to the movement they generate signals, and the signals are carried by the vestibular component of the vestibulocochlear nerve. This leaves us with the last component that was found by the otocyst, and these are the statoacoustic or vestibulocochlear ganglion. These are the cells that are made by breaking away from this otic vesicle, and their formation and patterning is very much influenced by the neural crest cells. So this ganglion further splits to form 2 further ganglion, the cochlear ganglion that goes inside the modulus and develops in the pillar in the center of the cochlear turns, and the vestibular ganglion that is present in the vestibular portion of the bony labyrinth near the otricle and saccule. So this brings us to the end of the inner ear. The otrical portion of the otic vesicle gives rise to endolymphatic duct. The saccular portion of the otrical vesicle gives rise to organ of Corti. Yes. And a fetus showing incomplete formation of cochlear duct with only one turn. Which structure is most likely underdeveloped? So it is a fancy way of asking from which structure does cochlear duct develop from. The answer is saccule. So let us move on to the middle ear. Middle ear is this tympanic cavity. This is a picture from Grant's Atlas, and it shows this pink area, that is the cavity of the tympanic, and on the lateral aspect there is tympanic membrane. On this aspect there is the oval and round windows connecting to the inner ear, and there are 3 bones in between. So remember this picture from the development of head and neck. Between first and second arch there is first pharyngeal pouch. So this is the pharyngeal pouch that is going to form the cavity of the middle ear. As it grows further, the connection in the beginning becomes narrow. This becomes the auditory or escutcheon tube, and this becomes the cavity of the tympanic cavity of the middle ear. So as it grows further, first pharyngeal pouch gives rise to this. So the cavity and lining of this means that in embryology the cavity and lining of all the cells of the middle ear, the lining will be endodermal in origin, as is coming from that. Same goes for the lining of the auditory tube. So it starts there, and it ends in this primitive tympanic cavity, and this recess gives rise to tympanic cavity. The proximal part remains narrow to form the auditory tube, and it usually remains in this state till the last trimester, in the 7th 8th month, where this cavity envelops, grows to free all these bones, the ossicles that are forming inside the mesenchyme. Let's talk about these ossicles. As you can very well see, the ossicles are developed in the mesenchyme before the cavity is developed. So these bones are developed from the pharyngeal arches. There are three bones: malleus, incus, and stapes. So the malleus and incus are developed from the first pharyngeal arch. So there will be neural crest in origin, and they will be developed in first pharyngeal arch. And the stapes part, it develops from the second pharyngeal arch, and they remain embedded in this mesenchyme as shown here till roughly the 7th or 8th month of intrauterine life, where this cavity gradually enlarges and ultimately it goes to its mature position, and that by doing that it lines whole of the medullary cavity, including the surfaces of these bones and the ligaments too. So the surrounding tissue dissolves by 8th month, the ossicles becomes free, and the epithelium surrounds and connects bones and the ligaments like mesentery-like fashion, and it includes all the lining of the medullary cavity, including the lining of the tympanic membrane. So here now you see that tympanic membrane has three origins. It has outer surface, that is the extension of the ectoderm. So outer surface will be ectodermal in origin, the inner surface will be endodermal in origin, coming from first pouch, and in between them it will be coming from mesenchyme. Okay, let us talk further. Muscles, there are two muscles. You can go to the lectures of gross anatomy of ear to head and better understand this picture, but let me give you a very quick preview. This is the posterior aspect, this is the superior aspect, this is the anterior aspect, and we are looking from the eardrum side. This is the sagittal section, and we are looking from the lateral aspect into the, this is the medial wall, and we are looking after removing the tympanic membrane. So as you can see, there are two muscles here. These muscles are this stapedius muscle that is getting attached to the stapedius bone, and then this tensor tympani muscle that is getting attached to the tympanic membrane. So as stapedius muscle was originating from the second arch, this the muscle is arising from the second arch too, and its nerve supply will be the same of, that is all the muscles of the second arch, that is the facial nerve. And this tensor tympani, as you can see, it is coming from the anterior wall and getting attached here and the malleus, handle of the malleus. It, as the malleus was originated from the first arch, this muscle also originates from the first arch and will be supplied by the nerve of the first arch, that is the trigeminal nerve. So it is supplied by the mandibular division of trigeminal nerve. So this is the tensor tympani getting attached here. So then comes the mastoid antrum. As you can see, this is the posterior wall of the middle ear. As you can see, there is it ends here and it remains a gap here. So this part, this is called the mastoid antrum. So this is formed during the later part of the fetal life, and it is from the dorsal expansion of the middle ear in the late embryonic life. But you have to keep in mind that mastoid air cells, so these are the mastoid air cells, the air filled cavities in the mastoid process. These mastoid air cells and the mastoid process itself are not present at birth. After birth, they develop as this cavity further expands out and making these air cells. Further, the pull of the sternocleidomastoid also helps facilitate this process, and the mastoid process, including the air cells, it get developed by roughly second year of life. So that's it, we are done with the middle ear. Let us check what we have learned by now. The histological section of the fetal ear tissue shows a cartilaginous shell forming vacuoles. Which spaces are formed at this stage? Cartilaginous shells are forming, and vacuole spaces are formed above and below the cochlea. So these will be the scala vestibuli and scala tympani, and among them scala tympani answer is given. The tensor tympani muscle derived from first pharyngeal arch is innervated by which nerve? It is developed by first arch, so it will have nerve of the first arch, that is the trigeminal nerve. Which structure derived from second pharyngeal arch articulates with incus? So malleus, incus, stapes. So malleus and incus both can be correct answer, but from the second pharyngeal arch. So that leaves us with the stapes answer. Now we are left to the last part, that is the external ear. It contains this pinna part and this meatus part that is cartilaginous in the outer one third, bony in the inner one third, and we will include also the external layer of the tympanic membrane. Meatus. Okay, here is the part where due to recent development the narrative is currently changing. A few years ago, and even still in most of the textbook, we have the explanation that the external meatus, it develops from the first pharyngeal cleft, and first pharyngeal cleft and the deepest part of the first pharyngeal cleft part makes the surface covering the tympanic membrane. But in the latest edition of Langman embryology, it says that it does not develop as from the first arch. Rather, it does not develop from the first arch, first, I'm sorry, cleft. Rather, here in the first arch, the skin becomes thickened and this invaginates, and then it gradually is absorbed to form the meatus. So this is a slightly change in narrative. So it is made of the invagination of the first arch as said by the Langman, but in the previous edition and still in the KLM edition, it is mentioned that it is made of the first pharyngeal cleft. Initially, this opening it's filled with a solid plug of cells, that is meatal plug, and it gradually dissolved by the 7th month. And when it is dissolved, that leaves with a lining of the ectoderm that ultimately develops into skin of the meatus and the outer side of the tympanic membrane, and that's it. So all of the meatus and the external part of the tympanic membrane are ectodermal in origin, and it continues to grow after the birth too, and it attains its adult length by 9 years of life. So that leaves us with the eardrum that has ultimately three origins: the outer ectodermal origin, the inner endodermal origin, and in between them there is mesoderm plus mesoderm in origin, depending upon the mesenchyme that was present between them. So this is also like thin skin. It is actually keratinized thin skin, and it is mucus layer with cuboidal epithelium, simple cuboidal epithelium. So that was the picture that we used earlier. And last but not the least, the auricles, the pinna. This is the first arch, this is the second arch, and between them there is the cleft, and then there develops the neural crystals develops these tags. So these are formed by the auricular hillocks. They are developed from the three on the this side on the first arch, then 4 5 6 on the other, and then they continue to go through, develop, and they form the shape of the pinna. And as you can see from the first arch, this anterior part, tragus part and anterior part of the concha is formed, and the posterior part, the major part of helix, antihelix, and antitragus, all of these are majorly formed of the second arch, and that explains the innervation. So the first and the anterior part is majorly supplied by the auriculotemporal nerve, and the posterior part it has some supply by facial and vagus nerve, but it is supplied by the branches of the cervical plexus. The first arch derivatives are supplied by the trigeminal nerve. A 12-year-old boy undergoes a surgical procedure to correct a congenital malformation of his right ear. He has microtia, small ear, a condition in which external ear is underdeveloped or absent. This malformation has occurred due to the abnormal development of ectodermal thickening from the otic vesicle, endodermal outgrowths on the first pharyngeal pouches, mesenchymal proliferation of the first and second arches, and neural crystals from the first and second arches. So mesenchymal proliferation can be a safer answer. This can also be a correct answer. But this can be a better answer because it's not purely mesenchymal in origin, it has mesodermal origin too. So tympanic membrane has a components from all of the following except it is made of ectoderm, endoderm, mesoderm, mesothelium. So it does not contain mesothelium. Mesothelium is the this the covering lining of the serous cavity including the pleura, pericardium and peritoneum. So a quick review of hearing. So when the sound waves travel from the external cortex meatus, they make the tympanic membrane vibrate, and they move the three ossicles and they vibrate here the oval window, and that gives this transmits the pressure waves into the scala vestibuli. And where this there is a natural frequency of the basilar membrane of the cochlea, and when there is natural frequency of a particular part of the cochlea matches with that of the incoming sound, that part of the cochlea vibrates, giving origin to the signals, and then the rest of the vibrations are dissipated through the round window. So I wanted to talk about impedance matching, because the vibrations traveling through air have to face less uh, resistance, because air molecules can be moved easier as compared to those of perilymph. So you the force of the vibration will have to be increased. So that is matched due to the change in sizes of the tympanic membrane versus foot plate. As you can see, the stapes foot plate is very smaller, and also the amplitude of the moving tympanic membrane is translated into a smaller amplitude of the stapes vibration, foot plate vibration, but that leads to smaller amplitude but higher force. So that is how impedance is matched, and the amplitude of the moving stapes generates the signals of loudness, and at which point does the natural frequency matches with the incoming sound, that gives signals to the pitch or frequency of the sound. Let's review a few clinical correlation. The congenital hearing loss can be there. It may be due to sensorineural or conductive causes. Sensorineural are due to hair cells of the organ of Corti, auditory of ganglia, and conductive may be anywhere from the external canal, eardrum, ear ossicles. It may be there. Then there is Tricker-Collie syndrome. We have discussed this in detail in the development of head and neck, and in that there is deficiencies in the development of first arch, and there are, among other issues that are being found, uh, facial abnormalities that are there. Regarding ear, we have external ear, meatal, and ossicle involvements. All are found because all of these structures involve the cooperation from the first pharyngeal arch. Down syndrome, it can cause conductive and sensorineural hearing losses too, and or sensorineural hearing too. Infections, rubella, cytomegalo, herpes simplex, they affect too. Regarding rubella or measles, keep in mind that this affects the eye too, and if it occurs before 7th week of embryonic development, that is more detrimental to the eye development, and if it occurs after that, it is more detrimental to the ear. Prematurity, maternal diabetes, retinoids, so all of these can have sensorineural hearing loss. Stapes development is very complex because of its dual origin. It is coming from the neural crest cell too and paraxial wisdom of that area too, and sometimes signals get mixed up, and the petrous part of the temporal bone, which is forming the oval window, remains fused with the foot plate of the stapes. When that occurs, the stapes cannot move against the oval window, and if it cannot move, it cannot transmit any signals inside. So there is sensory neural hearing loss, or say there is conductive hearing loss. And last but not the least, external ear defects. And external ear defects may be absence of ear, anosia, small ear, microtia, periauricular appendage, extra part, periauricular pits that are present, and they may not be anosia. Of course, it's a big problem, but all of these may not be a problem in itself. But these are mostly occurring due to some developmental issue of the neural crest cells. So the any abnormality to the ear should trigger the physician to check for presence of any other chromosomal abnormalities, because a lot of people with the ear abnormalities will have cardiac anomalies, facial abnormalities, because those things are also dependent upon neural crest cells. Thank you so much for listening. I hope so this lecture was helpful for you. If you have any questions, suggestion, or feedback, please let us know on our social media handles. Till next lecture, see you next time. Allah Hafiz.